Lesson 13.2, Ribosomes and Protein Synthesis. Our goal is to explain how and why the genetic code is universal and is common to almost all organisms. Our far side says, drive, George, drive. This one's got a coat hanger. All right, I hope you get it. Our objectives: identify the genetic code and explain how it's read, summarize the process of translation, and describe the central dogma of molecular, and it should say biology. Let's think about it first. How would you build a system to read messages that are coded in genes and then transcribed to RNA? Would you read the bases one at a time as if the code were a language, which is 4 words, 1 word per base? Or perhaps you would read them as individual letters that can be combined to spell longer words. Well, let's learn about it then. Our objective 1 is to identify the genetic code and then explain how it is read. The first step in decoding genetic messages is to transcribe a nucleotide base sequence from DNA to RNA. This transcribed information contains a code for making proteins. Proteins are made by joining amino acids together into long chains called polypeptides, and as many as 20 different amino acids are commonly found in polypeptides. The specific amino acids in a polypeptide and the order in which they are joined determine the properties of different proteins. The sequence of amino acids influences the shape of the protein, which, as we have learned, in turn determines its function. So the flow of information from gene to protein is based on a triplet code, a series of non-overlapping 3 nucleotide words. These triplets are the smallest units of uniform length that can code for all the amino acids. For example, AGT at a particular position on DNA strand results in the placement of an amino acid serine at the corresponding position of the polypeptide to be produced. Each 3-letter word in messenger RNA is known as a codon. A codon can consist of 3 consecutive bases that specify a single amino acid to be added to the polypeptide chain. And because there are 4 different bases in RNA, there is 64 possible 3-base codons. 4 times 4 times 4 equals 64 in the genetic code. This circular table shows the amino acid to which each of the 64 codons corresponds. To read a codon, start at the middle of the circle and then move outward. Most amino acids can be specified by more than 1 codon, though. For example, 6 different codons, UUA, UUG, and CC, CUU, CUC, CUA, and CUG all specify leucine. But only 1 codon, UGG, specifies tryptophan. The genetic code has something like punctuation marks. The methionine codon, AUG, right here, serves as an initiation or start codon for protein synthesis. Following the start codon, the messenger RNA is then read 3 bases at a time until it reaches one of 3 different stop codons, which ends the translation. So objective 1 is to identify the genetic code and then explain how it is read. Now you know. Genetic code is read 3 letters at a time so that each word is 3 bases long and corresponds to a single amino acid. Moving on to our second objective is to summarize the process of translation. So the sequence of nucleotide bases in a messenger RNA, let's call it mRNA from now on, molecule is a set of instructions that gives the order in which amino acids should be joined to produce a polypeptide. The forming of a protein requires the folding of 1 or more polypeptide chains. Remember? Ribosomes use the sequence of codons in the mRNA to assemble amino acids into polypeptide chains. The decoding of a mRNA message into a protein is a process known as translation. And this is how it works. Messenger RNA, or mRNA, is transcribed in the nucleus and then enters the cytoplasm for translation. Translation begins when a ribosome attaches to an mRNA molecule in the cytoplasm. All right, this is our ribosome right here. As the ribosome reaches each codon and mRNA, it directs or tells the tRNA, the transfer RNA, to bring specified amino acid into the ribosome. As you can see, it's happening here. One at a time, the ribosome then attaches each amino acid to the growing chain. Each transfer RNA molecule carries one kind of amino acid. In addition, each transfer RNA molecule has 3 unpaired bases, collectively called the anticodon, which is obviously complementary to the messenger codon, the mRNA. Okay? The transfer RNA molecule, the tRNA, for methionine has an anticodon UAC, as you can see here, which pairs with the methionine codon AUG. The ribosome has a second binding site for a transfer RNA molecule for the next codon. If the next codon is UUC, a transfer RNA molecule with AAG, anticodon, brings the amino acid phenylalanine into the ribosome. The ribosome helps form a polypeptide chain between the first and second amino acid. At the same time, all right, we've got these two, the bond holding the first transfer RNA molecule to its amino acid then is broken. That transfer RNA moves into a third binding site, all right, from which it exits the ribosome. The ribosome then moves to the third codon, where transfer RNA brings it the amino acid specified by that third codon. Over here now. The polypeptide chain continues to grow until the ribosome reaches what's called a stop codon on the messenger RNA molecule. When the ribosome reaches the stop codon, it releases both the newly formed polypeptide and the messenger RNA molecule, and thus completing the process of translation. So ribosomes are composed of roughly 80 proteins and 3 or 4 different rRNA molecules. These rRNA molecules help hold ribosome proteins in place and help locate the beginning of the messenger RNA message. They may even carry out the chemical reaction that joins the amino acids together. So, summarize the process of translation. Ribosomes use a sequence of codons and messenger RNA to assemble amino acids into polypeptide chains. Let's move on to objective 3. Describe the central dogma of molecular biology. So, most genes contain instructions for assembling proteins. Many proteins are enzymes which catalyze and regulate the chemical reactions which we learned about in Chapter 2. A gene that codes for an enzyme to produce pigment can control the color of a flower. Another gene produces proteins that regulate patterns of tissue growth in a leaf. And yet another may even trigger the female or male pattern of development in an embryo. Proteins are microscopic tools, each specifically designed to build or operate a component of a living cell. Molecular biology seeks to explain living organisms by studying them at the molecular level, using molecules like DNA and RNA. So the central dogma of molecular biology is that information is transferred from DNA to RNA and then to protein. There are many exceptions to this dogma, but it serves as a useful generalization that helps explain how genes work. Gene expression is the way in which DNA, RNA, and proteins are involved in putting genetic information to action in living cells. DNA carries information for specifying the traits of an organism. The cell uses the sequence of bases in DNA as a template for making messenger RNA. The codons in the messenger RNA specify the sequence of amino acids in a protein, so the proteins in turn play a key role in specifying the organism's traits. One of the most interesting discoveries in molecular biology is the near universal nature of the genetic code. Although some organisms show slight variations in amino acids assigned to particular codons, the code is always read 3 bases at a time and in the same direction. So despite their enormous diversity in form and function, living organisms display remarkable unity at life's most basic level, the molecular biology of gene. Our objective 3 was to be able to describe the central dogma of molecular biology. So the central dogma of molecular biology is that information is transferred from our DNA to RNA and then to proteins, which go and carry out their work, many times as enzymes. So objectives, again, are to identify the genetic code and explain how it is read, summarize the process of translation, and also to be able to describe the central dogma of molecular biology. All these objectives explain how and why genetic code is universal and is common to almost all organisms. And to finish up, there is your cute picture. Have a good day.